Hi dear students, today now we are going to talk about the theory of spontaneous generation. What is the theory of spontaneous generation? Theory of spontaneous generation is referred to as also the theory of abiogenesis. And we can see that there was a great debate on this particular topic, whether the spontaneous generation or the abiogenesis is true or the biogenesis is true. From the term itself, you can refer that abiogenesis. Genesis means the birth. So the birth from or the formation of living forms, if they are coming from non-living matter, that was the theory called the abiogenesis theory or the theory of spontaneous generation. Whereas biogenesis is a theory which states that life forms originate from living, pre-existing living forms. So there existed a great debate in the late 1660s regarding this because there was a lots of eminent people who stated that life forms originated from the dust as well as other various non-living matter. And there were a group of scientists who were supporting the spontaneous generation, but at the same time, there were a group of scientists who were opposing their theory of spontaneous generation. So, we are going to come across the various scientific evidences which were being laid forward in support as well as disregarding the theory of spontaneous generation. What is the abiogenetic theory first? The spontaneous generation mainly states that life originates from non-living matter. Aristotle said that any damp body could give rise to living things. And the main thing that was responsible for the origin of life was mainly the presence of pneuma or the vital heat. So, if the material contained a vital heat, he was saying that living matter could originate from any damp body. Damp means something wet. And coming to Herodotus, he was stating that living organisms could arise from non-living matter. And the suggestions that were being made was that crocodiles could arise from mud and life and so on like that. So, spontaneous generation is a belief that life can appear suddenly from non living matter without the benefit of the parents or any previously existing life. Now, if you go to think about that right now, we of course do know that life will only originate from pre existing cells. That is quite absurd to think that if you take some mud, you could make an animal. So that's quite absurd. So let's see what led us to the thought that life is indeed originated from living matter. And if you go to look into another science, there are lots of explanations of the spontaneous generation. Scientists who have been explaining them. And Van Helmont, he said that in the case of small animals only, this life originated from living matter. And he said that mice were being born from a shirt. Maggots could arise from meat, and mice could arise from feed. So, the first experiments to prove the spontaneous generation were done in the 17th century, and a doctor called Helmont, he declared that he performed a unique experiment. He placed a dirty shirt together with some wheat, and according to him, mice were born in 21 days later. According to the doctor, the sweat in the shirt was the active element which gave life to the inert matter. So, is that true? Is the origin of microbes the same? Is it from the non living matter? Now, from where do the cells come? If we cut a meat and it is being kept outside, we can see that it will putrefy and again it will begin to combine with the microbes and possibly with larger organisms like maggots. So, prior to the late 19th century, the origin of microorganisms in decaying matter was in question. Some maintained that microbes arose from other microbes that landed on the food from the air, and the others supported the hypothesis of spontaneous generation, which state that living organisms can arise spontaneously from non living matter. So, the theory of spontaneous generation was stating that let it be whatever it is, let it be a big form or let it be a small living form, all of them are being arising from non living matter. And the origin of microbes also they considered it was something like that, but that is not the true case as we all know. So let's just go explore it. The first person which we have to who conducted experiments against the spontaneous generation theory was Francisco Redi, and he was an Italian poet and physicist, and he tested the hypothesis of spontaneous generation by taking meat in three jars, okay, in three sets of jars. So three jars were open to the air. Always in every experiment we maintain triplicates. That is, for every reaction you will keep three sets. So he kept a set of three jars which were open to the air, and he found that maggots arrived in them. And he kept three jars sealed, and no maggots arised in them. And he also kept three jars with a fine net covering over that. Uh, in that also, maggots were not be present. So he concluded that the spontaneous generation theory did not still occur. Now, these are the three different sets which he had. So the meat has been taken in all the jars, and the first set of jars, the lid was open. So maggots happened to come from that. And the second one, it was being covered with a lid where no chance of maggots are there. And the third one, he put a what with a cloth-like netting material. So, what do we conclude from this particular thing, from this experiment? The According to spontaneous theory, what is it? Generation theory, we say that from the meat or the non-living meat arises the maggots. But when the jars were being closed, what happens? The maggots are not being formed in either the cases. When you cover it with a lid as well as along with the cloth-like material, what happens? The maggots are not coming and what infecting the meat. So it means that what happens? Something which is the microbes or something, the organisms which was responsible for the putrefaction of meat, as well as the maggots which normally arose from this, were in fact transferred from the air into the meat which has been kept into the jar. And when the jar was being closed, the maggots were not able to arise because they were completely sealed. So if you keep the meat alone, living matter is not being originated from it. That was the experiment done by Francisco Redi. And now the next scientist which we have is John Needham. John Needham said that the microbes spontaneously generated. And for this, the evidence that which he stated was that when you heated chicken soup and poured it into the flasks and covered the flasks, he found that microbes will appear. Now, normally we can see that when microbes grow, usually the broth or the liquid in which the microbes are being present are suspected, it becomes more turbid. That is, it won't be clear. So that's how you detect whether the microbes are being, or it becomes more cloudy. The microbial growth is in fact indicated by the cloudiness of the particular solution. So John Needham, what he did was he heated some chicken soup, and then after that he poured them into different flasks and covered the flasks. And after some days he found that these flasks which contained the chicken soup had changed into more cloudy forms. So that means that some microbes were being formed. So now he was heating the chicken soup in order to kill all the microbes if at all was present so that they would be killed. But he was saying that since all the microbes are being killed by heating, in spite of that, when they are being poured into the flasks, microbes were appeared. So he was approving the spontaneous generation theory. But is it true, or was there some loopholes in it? So let's just explore that. So there was some problems within his experimental design which he used here. So he is taking the chicken broth with microbial broth. So he is boiling it, and when he is boiling it, what happens? Everything is getting killed, and further it, he is covering it. Okay, and so what happens? The broth, he is finding that the microbes are growing further. Now, what are the loopholes which happened in John Needham's experiment? In Needham's experiment, he is boiling it, and later on, see, you just notice the thing. He poured it into the flasks, he heated the chicken soup and he poured it into the flasks, and then he covers the flasks. Okay, so the thing that has to be noted is if this thing is remaining covered and then he's simultaneously boiling it, there is no chance of something to enter into it. But what is he doing? After boiling, he's pouring into a flask and then covering it. So as a result of it, the broth is coming in contact with the air. So this is a loophole, and mm, it is in fact something wrong, and the spontaneous generation theory cannot be proved by this particular experiment. So later on, another scientist called Lazzaro Spallanzani, he said that Needham's broth got contaminated after heating. And for that, he did some experiments. He took some water and seeds and poured them into the flasks. And he covered the flasks using a cork. And then he heated the soup. One was being set for 5 minutes and one was being set for 1 hour. And he can see that the microbes did not appear in the 1 hour set, but microbes did appear in the 5 minutes set. So here in this case, Spallanzani was overcoming the loophole or the drawback which was there in Needham's experiment by covering the flask using a cork even when it was being heating. Okay, that is before heating itself he is covering it, and then he is killing all the microbes which could be present in the flask by heating it. And also he puts a time factor difference over here. The ones with which were heated only to 5 minutes, the microbes appeared. That is, insufficient heating could also not kill all the microbes. So if he heated it for 1 hour, the microbes are all killed. So these are the two different experiments appearing here. John Needham, he tried to prove the spontaneous generation theory. And what happens? The flaw in his experiment was that the flask was not sealed and it was exposed to the air. But in the case of Lazzaro Spallanzani, what he did? The boiled flasks of gravy to kill life, sealed one jar and left the jar. One was, of course, for all experiments, you should have a control. So when he sealed some flask, at the same time, he set another flask which was not sealed also. He wanted to check if this sealing is the something which was not responsible for the microbial contamination. So in the flasks which was not being sealed or which was left open, he found that microbes started growing. But the ones in which were being sealed using a cork, the microbial growth was not being present. So he concluded that the gravy did not produce a life, but the organism entered through the air. When the entry of microbes from the air was being prevented using a cork, the contamination or the growth of microorganisms in this gravy is not being present. So that's about Lazzaro Spallanzani, and he is disproving the spontaneous theory of generation. So that's what we are saying. Spallanzani took a large round bellied flask, I have already mentioned here, with tapering necks, and he then washed them thoroughly. He placed the seeds of various kinds in some and peas and almonds in the others and poured pure water into them. And he then melted the necks of the bottles shut because he feared a cork might not block the smallest of the microorganisms. So in some cases you can see the cork is being used, and even to make it little more sure, what happens? He melted the necks of the bottle so that even if the cock is, he was also scared because sometimes air could enter into it through the cock. And he then proceeded to boil one set for 1 hour and the other set for 5 minutes. And he made another duplicate set of flasks, which was sealed with corks that he boiled for an hour. And this was final set was Spallanzani's control group. Okay. After a few days, he went to check his results and he observed that single drops of each of the liquids under his microscope. He checked every drop under the microscope and he found that the sealed samples that had been boiled for 1 hour showed no signs of life, while the sealed bottles boiled for a few minutes and the corked bottles that were boiled for 1 hour and many had many observable microorganisms. So even in the corked bottles, if you're going to look, some amount of microbes were being present, but the ones which are being completely sealed from the air was being free from the microorganisms. And the time duration was also a factor. And thus, Spallanzani had proved that some microorganisms could survive in extreme heat, and more frequently that Needham's theory of little animals arising spontaneously was false. Then finally, we have another scientist who was saying about the spontaneous generation theory, and he said that production without a parent of a new living organism was being possible, and it was Pouchet in 1859. He also was talking about the spontaneous generation theory, and he always said that for a new living organism to obtain a solid organic substance just after the death was being needed, some life force was needed, and water was also needed. And he reported that the appearance of microorganism in a boiled hay infusions under mercury after exposure to artificially produced oxygen. And he said that the germs in the atmosphere air were not necessary to engender the life in the previously boiled liquids. Now, another scientist called Louis Pasteur, he began a polite private correspondence with Pouchet in which he suggested that Pouchet had unwittingly introduced a contaminated air. So, spontaneous generation theory was also being supported by Pouchet, but he also had some loopholes or some drawbacks in his experiments. And it was Louis Pasteur who found out that later on that Pouchet had unwittingly or purposefully introduced some contaminated air into his experiments when he was boiling the hay infusions under a mercury after exposure to artificially produced oxygen. So these are the cases of various scientists who were supporting the spontaneous theory of generation, as well as we saw few examples where the theory of spontaneous generation was also being disapproved. So we'll continue with the next session with some more experiments which are disproving the theory of spontaneous generation. Thank you for now.